that people to like see. So here we are in front of the world now doing these interviews, and I deeply thank each and every one of you for carving out the space to spend time in the room with us, learning about soil health and deepening into who you are and what your next steps on your own personal journey, individually and collectively. And today, in this hour and a half, we have the pleasure and honor of being in the room with Chris Trump, a Korean natural farmer, curious soul, lover of life, student of Master Cho's. He's also studied with Dr. Elaine Ingham with the Soul Food Web, and he teaches courses about Korean natural farming around the world, as well in person and online. So if you can't get to one of his live sessions, you can watch and learn from him on his YouTube channel, or you can go deeper into one of his courses that he has available on his website, which we'll share more with you about later. And today we are having a conversation about plant immunity through biodiversity and fungi, because Chris has been learning some new things as he's been going along and doing a lot of different tests, field tests, and um, personal experiments. And he's going to share them with us today and what he's learning and what we can apply to our own farming and gardening and soil health journey. So thank you for joining us, Mr. Chris Trump.
Thanks for having me, Natalie and Leslie, and setting this all up. And for those of you joining, I totally get being nervous to be on camera. It's a thing, it's real, but nobody's staring at you and judging you. You are just a part of this whole conversation. The only people being stared at are probably Natalie and I. So you can be uh, safe in your autonomy as one tile in many. But yeah, the faces are appreciated. Happy to be here with you guys and having a conversation.
Yeah, so Chris, I'm not even too sure where to start with this because I think we have just pop in the chat if you're brand new to Chris's work and brand new to Korean natural farming. Pop a 2 in the chat. So you're brand new, you never heard of Korean natural farming before, first time being introduced to Chris Trump. Um, okay, that's really helpful. All right. Um, New here, new here, new here. Okay, awesome. So we do have some people who are new. And we also have some people in the room I know that are very familiar with your work. So I think what would be helpful is maybe just to give a broad overview of the work that you're doing, and then we'll go deeper into it. We'll just bring people in. How does that sound?
Awesome. Yeah, I love it. We'll chat around natural farming. If it's all right with you guys, I'll stroll around a little bit while we talk, as it is peaceful and helpful for my brain at times. Um, I am, I'll share something fun that I haven't shared really publicly yet. I am standing in a spot where we will develop a learning center for agriculture, specifically natural farming. And my best friend is coming with his wife, and we're going to sit and just be here. I have a bunch of work to do. I'm harvesting some ginger and helping a client, coffee farmer down in Kona. But space and the I shared it with a shared all the sub projects, and they were like, "Wow, that's a lot." And so it will be a pretty ambitious, but in a very heart centric and human health as we return to land understanding about ourselves and land-based agriculture or land-based living. Um, so yeah, just wanted to share that. It will be something that will be shared online the work here. So even if you don't come and visit, which it'll be very open for that, you will be able to participate through remote um, remote access. Um, Natural farming is kind of what I'm known for. Uh, in some places in the US, it's called Korean natural farming because my teacher, um, uh, Master Cho, we call him Master. It's like the Japanese word sensei or teacher, or and it speaks of a teacher-student relationship. He was the creator of the technique that is kind of what I teach and learn about still from nature. Um, Natural farming is something that was before him, in that there's this Japanese iteration, and at the same time, Master Cho took these concepts and brought them together in an elegant kind of package, something that a farmer could take the whole picture of natural farming and apply it as a farming method. Um, and it's pretty great. And so I've farmed with it to scale. It was one of my early kind of learning journeys was how to scale natural farming and to scale it for our family farm, which is a 700-acre macadamia nut farm. It's about 2, 3 miles down the road that way, and now run by my brother. And yeah, wonderful place to um, get to practice natural farming as I began. Since then, where it's a type of agriculture I've gotten to participate in using in the wine grape industry, um, in turf management in the golf industry, in it's widely used in the cannabis industry for the plant benefits. And cut flowers. And I've been in Ireland a bunch working with the dairymen of Ireland, using grassland management and livestock. And it goes on. There's a lot of industries that in the last 14 years we've been able to kind of work directly with and seen a ton of success. And so those farms are slowly coming into their third, fourth, fifth years in operation with just ongoing success. And the kind of key points in natural farming is it's really about tending to the microbial life of your growing environment, returning it to microbially diverse, like healthy natural environment is. If you've seen the movie, it's kind of old. I'm dating myself, but Fern Gully, it's like this spot, and the fairies are flying around, and everybody's functioning symbiotically, interconnectively, and nature is so much like that. It really is um, a beautiful, complex world of interconnectivity and in symbiosis and diversity. So that is kind of the core philosophy of natural farming: is to partner with nature to grow food, to work with natural law and these really simple techniques of producing your own fertilizer and your own biodiverse amendments, which is one of the core biodiverse amendments is IMO, Indigenous Microorganism. We use some acronyms in natural farming. I apologize for that in advance. It is a little disorienting at first, like joining the military, I think, and getting a bunch of new words for things. But it's boiled down, it's a teachable, science-based, and solid method for growing food without going to the store to purchase your fertilizer or relying on pesticide and herbicide to protect your plants. There is a iteration of natural farming that my teacher, Master Cho's son, who I've also gone to Korea and learned from, has created, and that is called jadam. You often hear these words together. They are similar. And jadam, there is less of the fungal side of the soil food web, and there is, to make up for that, this incredible and really a gift to agriculture pesticidal suite. It's not really a pesticide because it's it's a um, it's a plant nutrient, and we don't really need to call it a pesticide because it puts it into another category altogether. But yeah, natural farming is this beautiful kind of accessible way to grow crops that are full of nutrients, nutrient dense, and lacking any things that humans shouldn't eat. So that's yeah,
Yeah, thanks for that overview, Chris. So
totally.
so for example, we're fermenting different plants and different um substances like sugar and beer and different things we're putting together and fermenting and making solutions for them, which are nutrients for the plants, to put it in another type of language. So
Hmm.
um and Chris, can you give us just a snapshot of what IMO is and how that's used? Because you've talked a lot about how IMO is probably one of the kings when it comes to um, microbes and or maybe queens when it comes to nourishing the soil and the environment. So can you tell us what that is and why that is and how to make it and how it's connected to what you're learning about now with the pest
Yeah,
disease
absolutely.
management?
So one of the things we'll talk about today is something we developed over the last 10 years, so just trial and playing with, called IPMO, integrate indigenous predatory microorganism, which can help us deal with bugs in a natural or just like nature does it kind of way. But IMO is stands for indigenous microorganism. We have all heard about the microbes we can buy in jugs for our farm. Incredible tools for tending to the nutrients we need for our plants. But IMO works with the tree and the soil in a process that James White named rhizophagy. And basically, the any plant or tree like this one, as big as it is above, is full root base below ground that looks very similar to the above ground tree or plant. And those roots down below are very similar to a human gut, whereas a human gut is tubular, and inside we have our dirt and nutrients and microbes that process our nutrients for the human. The root is rod shaped, and right around or outside of it, it has the food, the dirt, the nutrients, and the microbes necessary to process those nutrients. Um, in a healthy natural system, the the tree is getting incredible on-demand access to nutrients through biochemical communication. Basically, the tree is photosynthesizing and producing sugars and excess nutrients put out around its roots, and those are picked up by fungi and bacteria, archaea, yeast, the whole of the soil food web, and that causes the microbial community to thrive and to grow and to increase. One of the key players in that is fungi, as they are like the subway system and the bone structure all at the same time. And that those microbes can process fungi, can process the dirt, the sand, silt, and clay, the stuff that you know um, that becomes ready minerals for the trees. Um, what IMO is a very effective way to go to the healthiest places, the fern gullies of the local environment, and cultivate diversity, real diversity, testable um, diversity, and get it from these wild places that have been pretty untouched by agricultural systems or human interaction, and bring them back to our growing environment. So a typical farm has been tilled, processed, used for generations. And as a result, much of that work, though not inherently negative, has diminished the microbial diversity. It just happens. It's part of the outcome. And what we can do in natural farming is we can go and coax microbes into. We put out rice boxes. Rice is used because it is high in fat and protein, which are fungal foods. So it actually gets cultured by fungi, and we found pretty amazing diversity as far as the fungi goes. We've gotten to do some DNA testing. We really do need to get more of that accessible for the science-minded to read, as it's right now a lot of within the community and not published. But we go and we get incredible diversity to culture on rice. We put it in a dormant state using sugar. The dryness of sugar is like the dry salt barrel that they would put fish in for a long sea voyage. It causes everything to kind of go dormant and stop. Um, and then we grow it out on grains and wood and liquid nutrients. And so in the end, we've gotten a little snapshot of natural diversity from a mountain place or a kind of a wild, untouched zone, and we get this large kind of usable material to re-inoculate our farmland with as a result. And it's a bit nuanced. There's some details that you have to follow, but it is a accessible means of creating a product that we don't have access to on the shelf. There's not really a diverse microbial inoculum that we can buy. So this per gallon of dry material is probably the comparable market value would be $500 to $600 for a gallon of IMO4. And we couldn't even buy that if we wanted to. Um, and so it's the best of the microbially diverse products you can make, um, unless you have a year or more to make it. If you had a static pile on the edge of a wild forest, you could create a really amazingly diverse microbial community. But IMO plays this amazing line in the nuance of fungal development that allows you to culture a large amount of diverse microbes in 3 to 4 weeks.
So fast, so powerful. And Chris, with the IMO, tell us how you've scaled that. Like what I mean, lots of backyard gardeners are practicing it, and but what to what scale have you been able to use the IMO?
Yeah, I was making um, really rich 90,000 pound piles for our farm here in Hawaii when I was doing it in solid state form. I then kind of moved to a liquid version where we trialed a bunch of different ways to get diversity to grow out in liquid form. And so I'm working with one of the largest Kona coffee farm in Hawaii nowadays, and they're using a liquid version of IMO. which has mitigated their pest and disease problems. So they were dealing with coffee borer, they were dealing with rusts and leaf blights, and they have 100% remediation now by applying this diverse liquid version. That's much cheaper, um, per acre, but it isn't as instantly effective. You don't have as much soil microbe development um, kind of instantly like a solid application. So it requires reapplication to kind of keep protecting that canopy and over time developing the microbial diversity in the soil. But yeah, pennies on the dollar compared to solid application. The solid application is still cheap. Um, I taught this, I mean, it as far as scale, it's scaled in several different ways. I got to go to Burma, Myanmar, and teach a community of 1,000 coffee farmers who are all very smallholder, everything by hand on their farm, coffee farmers. But there's 1,000 of them feeding this one um, coffee buyer who is there really to provide the best wages for these coffee farmers. Really kind of sweethearted business model they have there. But we went, taught this community. They all went out and taught their friends, and now there's a whole region in northern Myanmar that uses natural farming successfully. We taught them basic composting using those same techniques, and so that is a form of scaling a whole region being able to convert from buying that $100 a bag fertilizer from China and able to shift to a more sustainable and safe, meaning they're not having as much crop failure um,
Mhm.
type of agriculture. I've also worked with my friend Kyle Schnell out in Iowa, and we did a project for his farm. It's 1,000 acres. He's this sixth generation row crop farmer there in Iowa, and we made a large pile IMO. We had 1,600-gallon tea brewers, and he was putting it into his large cedar combine, injecting liquid IMO and FAA plus nutrient into the ground with his seeds. So that was definitely large scale, and a bunch of kind of scales in between. I was just out in Bakersfield. There's a um, award-winning olive farm making great olive oil. They also do pistachios and almonds, about 350 acres there in Bakersfield. And they have a liquid IMO production lab now where they make small batches of IMO and spray out their 350 acres using these tea brewers and
Mm.
air blast sprayers. So yeah,
So
it's
cool.
it's definitely scalable, and that's kind of the goal is to see how America's kind of large scale agriculture shift into a sustainable way to grow food.
Yeah, yes, amen to that. And tell us about so the liquid IMO though, are you taking that IMO that you're making and then you're turning that into like a compost tea or an
That's exactly right.
extract? Yeah, okay.
Yeah, so not an extract. I think there's great merit to extract
Hmm.
in that when you have a good um, microbially diverse compost, you can just wash it and basically suspend. You're not creating anything new. You're just taking out the microbes, suspending them in water for easy application, and
Hmm.
that way you can get it onto the leaves of the plants.
Hmm.
um, but an extract is only as um, there's only as much quantity present of microbes as there was in the original compost. So applying that compost to the soil, you would still be getting that same amount of microbial life out. You wouldn't be necessarily multiplying it, which there's nothing wrong with that. It's a way of doing it. What we've found, again, because of just finding that sweet spot of nutrients and balanced food, for them to grow in a water column with some aeration and this food mix, we can cause a handful of IMO to turn into 40 gallons of a whole lot more um, quantity of microbes than we started with. So we can have a small amount as an inoculum to the water column and then grow out a large quantity, get it applied to the canopy and to the ground. Um, but that's something that the regenerative community in agriculture has actually struggled a bit to get a truly diverse liquid compost. So actively aerated compost C is something people do. It's done all over to varying levels of success. And so um, what I really like to point out when we talk about actively aerated compost tea, AACT, which is kind of the standard term for this. We call it liquid IMO, but it is in effect very similar um, to actively aerated compost tea. Um, People make tea for different reasons. Um, there's two categories I like to put them in. Either nutrients, where we're putting some microbes and some nutrients into a water column to further break down the nutrients and make them more plant available during the brewing process. So that's a nutrient tea. Um, Typically, nutrient tea so full of nutrients that there are specific microbes, often lactobacilli family microbes, that are breaking down those nutrients, making them more plant available. And fungi, the mix of nutrients and enzymes, the pH, all of it isn't quite right for fungal growth. Whereas the other category of actively aerated compost tea is more like a specifically aimed at microbial development and not so much a food. It's more an inoculum. Food can come later, food can go on the ground, but we're really looking to kind of restore the gut flora, if you will, of the plant as the main purpose of the tea. And that will have less focus on what nutrients are in there and more focus on what balance of materials, nutrients, and such are best for growing out microbes in a water column. And so figuring out your why on your tea, I think, is important, and you'll fall under those two categories. Um, Both have great purpose, but are definitely different in kind of setup and in outcome.
I love how you pulled that apart for us, Chris. Thank you. That's really, really
Yeah,
helpful. Now tell us about IPMO and what you've been
yeah,
discovering with it and what it is and what's exciting you about it.
yeah. Really fun. Has anybody heard of IPMO in the chat? Um, And has anybody messed? Maybe raise your hand if you've, or type a me if you've messed with compost teas or actively aerated teas. Or has anybody played with this?
So me, if you played with P, played with P and T. Sorry. Oh my gosh, I think I'm funny. Oh, okay, so me, if you've played with tea, and but also just say yes if you have heard of IPMO, which is different. So you say yes if you've heard of IPMO. All right, we got a couple yeses in the
Awesome.
house. Awesome. And just a one if you're brand new to all of this. Just a one if you're brand new to
Well,
all.
we have some newbies. We're bringing
Yeah,
everybody along together. I think that's the goal.
awesome.
Um, so yeah, extract is like I said, it's pretty safe. You've the thing that I don't love when I'm trying to scale and cover a large amount of area, the reason I don't lean to extract is I could already apply my IMO in a solid state and get those same microbes onto my plants. And that solid state application is really effective. So um, that's why I wanted to see what we could do as far as multiplying out microbes in a water column, and we found a sweet spot. So I strongly encourage people, if you want to play with that, to really hone in on the exact recipe, as it is the sweet spot in production in a water column. But um, I'm really excited to talk about IPMO. I'm really excited to share it. It's been a long time in process. I do um, don't share things quickly before I trial them. That is a strong ethos of mine. I learned natural farming in 2009, and played with it more and more and started scaling it in 2011. And I never taught it until 2014 is the first time I taught, because for me it was really a flower.
Mm.
Um, it was really important to know its efficacy. People really excited about it. Natural farming, they were making a nonprofit, and they're like, "Hey, do you want to participate and join the nonprofit and do all these things to further this?" I'm like, "Well, I'm gonna just go farm with it to see how it works." And I spent 4 years trialing. We did a 14 tree trial to a 5 acre trial with some significant year to a 144 acre trial over 4 years, running that while I was beginning to teach it. And I knew what the ins and outs and uh, how to help people avoid some of the pitfalls I did. So IPMO is not something that we're pulling out of an idea jar quickly. It's something that's now been trialed by um, wonderful science-minded agricultural professionals for over 10 years. So this is something figured out a long time ago, and we've been with. And it, and we found this is very much one of those kind of um, the process of culturing
Chris, we're just losing you a little bit. Is he cutting out for anybody else?
other
Yeah.
in the carapace.
Chris,
So
can you
it's
hear me?
yeah,
Okay,
yeah, I
you're
can hear you.
cutting out. We couldn't quite hear you.
Oh, sorry, I'll stop walking around.
Yeah,
Um,
now we can hear you again.
okay, my bad. Um, I went into
So we
the
heard we heard that you trialed it and that it's been 10 years in trial and now you're really excited to share it. That's kind of where we got.
yeah, yeah. So this is you know, just for context, because some people are like, what does it take to do all this? I've never heard of these things. Like, what kind of equipment do I need? For example, if somebody wanted to do this and learn this on their farm, it would be a small covered space with a dirt floor, would be how you could go through the whole IMO process, making it. It's like turning a small pile of compost. It just has specific parameters. Liquid IMO is a little mesh paint strainer from Home Depot and a small, $30, $40 air pump, and some nutrients, along with some inoculum, and 24 to 36 hours to brew it. We're talking about pretty simple tools. The hardest aspect of things in natural farming is really just the commitment to a little bit of time to learn, just like learning how to make sourdough effectively, or if you want to cook meringues, it's kind of like learning baking, or a new riff on the guitar. It's definitely just takes a little bit of learning curve. Um, so IPMO is similar. We're not talking about a ton of tough tech. We take rice Again, we use white rice. The um, though we talk about indigenous microorganisms, and we are focused on culturing indigenous things, we're um, we're okay using rice because it's one of the most effective ways to culture fungi kind of artificially, meaning you're not just going out into the forest and getting a stick pile and bringing it back and it has fungi on it. We're intentionally getting it to grow on a substrate. Um, Rice has been bleached washed if it's white, so it's been compromised a little bit in its molecular structure, which allows it to be a really ready food for fungi. It allows us to do things slightly faster than the slow chew onto some wood or whatnot. So, what we do with IPMO is we add beetles or insect frass or your local problem pest. So, I'm working with Ginger here in Hawaii on our farm, and one of our main pests is a Japanese rose beetle, little tiny beetle. So I go out at night, collect them off the leaves, and I put them in some rice that I've cooked. Um, and I put that out in nature with some fungi around it. I find a fungal rich environment, and I get, not every time, but every other time, um, I'll get blooms on the beetles themselves, these white, puffy microbial blooms. And I have just found a carapace eating fungi. So when I first learned about IPMO, I learned about it from nature. Nature taught me about it. I was doing IMO. I was making 500 gallons of tea at a time. We had a whole streamlined process for our farm to be able to spray out liquid IMO on our macadamia nut trees. And the substrate I was going to make my material on, this highly fungal diverse inoculum, had weevils infesting it. When I went to get it to collect and put on our to make some more IMO for, and I was bummed because I was like, oh no, I have to go get more material. This is terrible. It's going to take me a week. I'm in Hawaii. Everything's hard to get. And I decided to make it anyway. So I made my IMO on weevil infested grain. And while I was doing it, which I had done this many, many times, so I had very clear parameters of what was going in and what to expect. And I was getting unique or never before seen puffballs of white. And I'm looking at the center of these puffballs, and there's little weevils in the middle of these white puffballs. And I was like, that's really interesting, kind of strange. And I had all these questions, but I was busy. I was managing the farm. So I made it into liquid IMO, sprayed it on 20 acres, and like a good farmer, walked through my 20 acres to see, not specifically because of the beetles, but just because that's what you do when you get an application of fertilizer out. We're checking how our trees are responding. And as I'm walking through that orchard, I see puffballs in the crook of a branch or on the side of a tree. I'm like, what is that? And I look closer and I have a beetle that's covered in white fungi and white bloom. And I'm like, oh, and where I didn't have time before because I was busy, now my curiosity was way, way higher priority than the work I had to do. So I ran down and started to look into, okay, what eats beetles? What's going on here? And what I found was in my research was that the predators of my pests, the main pests I have in my crop, are fungi. The leading predators of a lot of our plant pests are microbes. And so what I was seeing was an entomopathogenic fungi, a fungi that eats bugs. Um, and this was like, oh my gosh, how wonderful. Now we have an indigenously cultured. I cultured those microbes within a few miles of my farm all around. So up in remote spaces, so those fungi don't travel easily those several miles. But now I had reestablished them on our farmland, and they were able to have pressure against my pests. Um, this became a significant curiosity, and I had some friends run into locust problems in Zimbabwe and other beetles in other crops in different places in the world. And over time, we played with this and found that it was completely repeatable. We could, in different contexts, in different place, we could buy even commercially available insect frass, which has plant parts in it, and add that to the rice, and we would get the same results. And so by putting out in these wired environments food for entomopathogenic fungi, we can culture, wildcraft our own predatory fungi and then reestablish them on our gardens for some degree of perpetual protection. Um, we can also use it as a foliar spray to knock down bug pressure. So this is something now that is being used to great success by a bunch of people just watching the video on how to do it and going out and doing it. And so um, the amazing thing that we see in plants is how incredibly resilient they can be if they have living soil in their root zone. If they're getting that diverse nutrition, they're able to produce complete proteins in their cell walls and really protect themselves from bugs, which I can explain more on. But yeah, it's IPMO is this tool that is open source information and costs nearly nothing to get control that is more than we have with commercial products at this point. So really, really enjoying playing with that and seeing results across industries that are pretty fun and impressive. So we're actually beginning a trial with University of Hawaii and a doctor friend of mine. We're going to try and get us a white paper for the community to use and show the efficacy here in the next year.
It's amazing, Chris. It's so curious. I love there's I mean there's so many parts of that that are awesome. Um there was a question that came up, and that is so for example, if you have a problem with flea beetles in your potato field,
Mhm.
if you would recommend to catch a flea beetles and put them in a pot of rice and use that as an inoculant to draw the fungi and help control the flea beetles, or is there a process? Like how important is the I is putting the rice out into an old growth forest or in, how important is that part?
So the only so it's not magic. We have to remember this that like this is something there's a stages of process that made it work, right? And so what can we do? People want to make it make really anything simpler all the time. That's our bent as humans. We want to make it more accessible and simpler. What we have to question is when did we get the necessary things along this process that I'm describing, that I have a video explaining? Um, what are the necessary steps? What if we take that one step away, which step would make it so it doesn't work? And so if you just took that beetle and you put it in some rice and then you waited, you didn't put it out into a wild environment where there's a bunch of fungi, the only way that you're going to have an entomopathogenic fungi grow in that environment is if it's already infested that beetle that you put in there and it was on it before you started. And then yes, maybe it could grow further on that, but if that was the case, you might not have as much beetle problems. And so it is more likely that you're going to have an undisturbed or healthy entomopathogenic fungi in a place where it's naturally being controlled, where you don't have a beetle infestation, right? We have these infestations and these out of control populations due to a die off in diversity, die off in the natural checks and balances that nature provides. And so by going into these places where, I mean, you find I go on these nature walks and I find beetles with covered in fungi and fuzz, you know, and some of them are, you know, just cleaning up the forest floor. They're just eating what's there. But if they're eating carapace, mostly they can cause problems for a living beetle. So it is important to go out into some degree of a wild environment and really given access to fungi, because if you don't have control already, if you're not seeing in your garden infested beetles with fuzz on them, they're not going to be um, it's not going to be appearing out of nowhere. We have to find it somewhere. And so I also encourage people, they're doing the whole protocol, they're going out and they're putting it in the forest, and then they get bloom in their rice, but they're not seeing any bloom on the beetles that they've put in the rice. That doesn't necessarily mean you have some IPMO or a predatory fungi that was cultured in that rice. Maybe you have just normal IMO. You have fungi that likes to eat fats and proteins, which is great, but really encourage people to look for that bloom on the carapace itself to see if you have actually cultured something that might eat your insects.
Mm, good insights and good question. And so it sounds like, Chris, if I have pressure insect pressure, then and I want to make some IMO to make IPMO, then I might not want to just put one little box of rice out.
Box
I might
out.
want to put out five in five different areas and see and hope that one of
Absolutely.
those bloom on
Yeah, that's right on the nose. It's a bit of a numbers game.
hmm
Um, we're you know, it's kind of like macro mushroom hunting, right? You go mushroom hunting and you're
Yeah,
like, oh, there's that one spot that all those mushrooms grow. That doesn't mean that if you go to every forest in your area that you're going to find morels.
that's right.
Morels are there and there and there. Same thing with entomopathogenic fungi. They're not going to be in every location in abundance. And so looking for spots with a bit abundant visible fungi and then hoping for that entomopathogenic fungi is part of the journey. You're looking for your best mushroom harvesting spots, and it's just like macro mushrooms. Not every spot is ideal for that.
So what are we looking for when we're placing these little IMO boxes out and IPMO boxes out in the forest floor?
So with regular IMO, we really want to see the visible individual strands of fungi. If you can see an individual visible strand, then it is at least 4 micrometers in diameter, because then naked eye, the human eye, um, sees anything smaller than 4 micrometers as color or fuzz, not as an individual strand. So we don't perceive things smaller than 4 micrometers with the naked eye. So most of your disease-causing fungi are 1.5 micrometers or smaller. So if we're seeing those individual strands, we're seeing the beneficial side of the fungal community, the microscopic fungal community. Um, and then as far as IPMO, there's this incredible thing that happens with seed development, germination. Every seed, as it germinates, for a short time in its germination, right after the little new root emerges from the shape of the seed, it is full of chitinase protein. It's full of the same protein found in the chitin or the exoskeleton of bugs. And what I believe about that is it's actually putting out this protein to create initial relationships in its new forming root zone with beneficial fungi that will protect it from bugs. So it's taking the protein that the fungi would look for when the entomopathic fungi, they look for in insects, and it's putting that in a short phase of its life, it's putting that into its root zone so that the entomopathic fungi say, I'm going to hang out here, I'm going to create relationship with this plant and protect it because it's one of my food sources, and it creates symbiosis. So this is a theory, it's not widely tested, but malting grain produces high quantities of um, chitinase protein, which is what entomopathogenic fungi are looking for as they eat these insects. And so going to a place where there's maybe a bunch of pine seeds, or a tree that's producing other kind of nuts or seeds that are germinating on the ground, that might be a place where you'd find a higher abundance of entomopathogenic fungi, because there's the protein that they prefer in their diet. So I'm always looking for, we go out and here in Hawaii, I'm looking for a kukui nut tree that's putting out a bunch of seeds. I'm looking for, there's these little pine cones that, mini little pine cones. I forget which, I think it's a ironwood or a false ironwood pine tree produces these little pine cones with seeds in it. You'll see the seeds germinating, and you'll see all these mushrooms coming out of where the seeds are. Two reasons. One, seeds are high in fat and protein, so any fungi is going to like it. Two, a germinating seed is going to have the chitinase protein. So nerdy reasons to look for seeds in the wild, but really, we're following the breadcrumbs that nature puts out. I learned this from nature. Nature showed me that there was something eating my weevils that also is eating the green stink bug that is my main pest in my trees. And I followed the breadcrumbs, and we now have a technique that is a way for any farmer with limited resource, limited cost, to wildcraft cutting edge or top quality insect control.
And what do you see as the biggest hurdle, biggest challenge when people are trying to make IPMO? Where are they misstepping?
Um, that's a great question, a really important one. I want to address a real quick chat question,
Yeah.
and then get to that. I think it'll be um, complimentary. Somebody asked, "What about your beneficial insects?" Um, Great question. Um, one of the things we did trials with are with pollinators, with excuse me, with bees, and bees are not susceptible to this material unless they're sprayed directly. So a great way to apply IPMO and not affect your bees is there's times of the day that the bees are not out on your trees, on your plants. That would be the time to apply your IPMO. Look at the bees eating schedule in your environment, and you can ask the local beekeeper, when are the bees out? When are they in their hive? And just spray it when they're in the hive. After it's been sprayed, the bees actually benefit from these fungi. They will use them in their hive. They will use them in the soil below their hive to protect themselves from like hive beetles and other things. So bees thrive in microbially rich environments. They have a whole process of cleaning themselves when they step on something. But yeah, we do find that if you completely blast them with a full spray, that you can kill a bee with a IPMO treatment. So do be aware. Um, However, it's not going to kill the hive. It's not like they're going to go back and they have a whole cleaning process. And yeah, so you will kill a few bees if you blast them directly. But if the bees show up there the next day and land on it, it doesn't hurt them.
Mhm.
This has been uh, many environments we've gotten to trial this with, and really observing the potential risks. So
Mhm.
again, back to the efficacy trials being really important.
Yeah, that'd be interesting too. I wonder what it would be to, the mites that are in the beehives and make an IPMO with that and then put some water out that the bees might like to drink.
This is
Pretty
this is
fascinating.
uh this is the way to go. Treat the below your hives with IPMO and protect your bees. So we have uh, we had 140 hives. I think we're down to like 90 hives because we just haven't had it be as much of a priority in the last few years. But we buy the equipment and we have a beekeeper. He's a queen breeder here in Hawaii that manages our hives. And he's always talking about how our hives are always splitting and we're getting an abundance of bee production and we don't get the diseases. And so while he moves other hives around the island, he takes bees continually from our hives as the fill back up his other hives because our organic macadamia nut orchard they're thriving. They don't get we have a hive beetle here in Hawaii, and we have the varroa mite, and yeah, both are things they're not susceptible to in our particular hives. So I attribute that to treating all the hives with IMO under the hives, and that just being kind of their um, kind of natural protection.
Very cool. So how do it's just really quickly, we are going to put some links in the chat and we'll also put them on the watch page for Chris's YouTube videos on how to make IMO and IPMO so that you have those resources. And then we'll also link his website as well, where he has a lot of interesting sources and resources. And so to learn actually how to do it, that's probably one of the best places to go is directly to his courses and or his YouTube channels. And then we're gonna do our best to get the other questions answered in the chat. So so Chris, what are the ways that the most common ways people misstep in making IPMO and how can we make sure we pay attention to those and mitigate them?
Um, yeah, so I think one of the ways that IPMO can be one of the ways you could fail is using too much insect frass when making your rice. So, or too much of the insects. So, insect frass and whole insects are both pretty high in nitrogen. And so you got this rice, you're cooking it. Now you're cooking it with insects. And if you have half quantity insects, half quantity rice, you're going to have so much nitrogen in there that it's kind of like making like a manure compost. You're going to put that out, and even in that week time, you're going to have this like thermophilic process start that will start a decomposition. Um, and that's not what we want to have happen in the IPMO production. So a handful in a full pot of rice would be enough, or even two handfuls, but not anywhere near 50%, not even like 25% would be the absolute ceiling max of insect frass or insects in your rice. Really, you need 10%, 5%, something like that. It's not a whole lot. Um, so yeah, don't overdo it with the insects in the rice process. Um, the rice making, the box itself that you put the rice out in, um, these are pretty important or detail specific processes. In my video on IPMO, I give a direct, like a specific ratio of water to rice for a rice cooker to make kind of good al dente rice. You don't want to cook your rice sticky so that it's one big conglomeration. You want airflow through it. So the rice grains holding their individual shape actually is really important because the main purpose is cultivating fungi. In natural farming, beneficial bacteria is very easy to accomplish. We're doing that throughout the whole process. But that aerobic big fungi, that's harder to get to, and so we want to care for them. And aerobic rice is created by not overcooking the rice. Um, the other thing is we want any container we're putting our rice out in to be Swiss cheese, full of holes, just like a strainer for pasta. We want all the airflow to come up through the bottom and through the sides. And so if you're making a cedar box or you have a basket, we just want to make sure it has enough airflow that when you put it down on that fungally rich soil where you see those individual strands, um, a spore that gets stirred up is going to hit rice. Some rice is falling out, and the fungi is using it like a stairway of heaven to get up into the rice box. We want so many holes that a little bit of our rice is falling out if we move it too much. Um, and those are some of the most important details. Um, and yeah, more videos is something that I'm working on so we can continue to just make easy access. But there are great videos available on YouTube and information on the website and communities that'll talk about it with you in the online space. So we definitely want to answer those questions. I even field specifically about IMO. I field IMO questions on my Instagram when somebody sends me a picture of their IMO or sends me something, I answer every single IMO question. I might not answer every single farm question or theoretical question that comes my way, but I will answer every IMO question because I know there's a little bit of a learning curve. Um, yeah,
Bless you.
those are some of the main or most important details.
Thank you. Thanks for that, Chris. Um, Greg says they've been having experiencing long drought periods in northern Great Plains, so they don't tend to use ground applied liquid IMO as it seems they should be soaking this into the ground, and this doesn't happen in extended periods of drought. Oh, do you have any insights around that?
Yeah, that's a good question. So in a place where there's low water, um, and you're culturing IMO, you're going to be finding what blooms on your rice is something that has been able to persist through the drought in your environment. Um, and so, one of the best things you can do, the most effective things you can do, though it's a little more expensive than a liquid IMO, is to make solid state IMO, apply it, and then put a light layer of mulch on it. Is a major undertaking depending on your scale. So definitely understand that. Uh, JMS can be used, Jadam microbial solution, which is easy to make. I have a video on my YouTube. Just search Chris Trump JMS. Um, that is something that you can water in and you dilute it 1 to 20. So there's that 20 parts water carries it deeper into the soil. That can be really effective. But yeah, using indigenous microbes in a arid environment is the key to having microbial growth in an arid environment. There are already the microbes that are still persisting even through the drought. That's why they bloomed in your rice box. That's why you could find them in the first place. So I do some farming in Boise, Idaho, and it's high desert, and we find even in 105-degree summer days, we're finding blooms of indigenous fungi and able to get it back out to the croplands. So
Very cool.
keep trying.
Here's a good question, and I'm going to add to the question. So it's Mark asks if apply so would adding a supply of brewed IMO or made IMO to a supply of biologically complete compost make a difference? So could you add IMO or IPMO to a compost pile and leave that sit for a while to have that move through it? And
Yeah, so I don't know the makeup of BioComplete compost. I know it's being touted as everything a plant needs. So I can't answer specifically to that. I'm sorry. But if you want to take a batch of IMO and grow it out on a compost pile, one of the best ways to do that would be a static pile, which has like a 70 plus percent carbon makeup. So you're going to have plenty of aerobic spaces for fungi to grow. Um, Taking IMO inoculum like an IMO2 and putting it in anything thermophilic can be a way to really just lose your diversity. So you do, yes, absolutely. You can take some IMO, IMO2. We can even make liquid IMO out of IMO2 and just use that straight on the plants. But taking IMO2, um, growing it out in a liquid IMO and spraying it on a static pile is a great way to further your inoculum. Or, yeah, taking finished IMO and applying even just a handful to a decent size, hand turn pile is a great way. But the best I would say would be static pile, inoculating it and letting it take its time and grow. You're really going to see, especially with that high carbon amount.
Mhm, mhm. Um,
Yeah.
Chris Greg says he's watched your videos on making compost tea from IMO4, and he's wondering to make the value of IMO4 more instantly available. Have you tried making liquid extract from the IMO4 so you don't have to
Yeah.
wait the 24 hours to brew it?
Yeah. So extract is great. The reason I encourage liquid IMO is because in, you know, 14 years of playing with it, I'm able to get this massive increase in fungal biomass. I'm able to get I start with this amount of fungi and I grew it, 4,000x as far as biomass goes, or, you know, that is a very random number. I'm sorry, I don't know the exact X amount. But whereas extract is great, you're washing it with extract. What you're doing is um, you're making coffee out of coffee grinds. You're not multiplying the amount of coffee that was in the original grinds. You're actually leaving some coffee behind, but you're getting a fast available form of the coffee in the grinds. Same thing with an extract. You're taking IMO, there's a bunch of fungal spores in there if you made your IMO right, and you run water through it and you're washing all those spores into water. And now you have, instead of a solid bag of microscopic marbles in your IMO4 powder or material. Now you have washed a lot of those marbles into water column, and now you're going to splash them out, which is great because it gives you a way to move them. But if you take that same material, that handful of IMO, and just spread it really thin, which is like 330 pounds per quarter acre, that's our IMO4 application. That's like it's literally a dusting, like you might put out lighter than you put out diatomaceous earth for like protecting from bugs. It's really a real light dusting, and all you're doing is taking those same fungal spores and getting them seeded on the ground, kind of like cover crop seed. So whether you wash it for an extract and put them out in water, or you throw it out by hand and just water it in. Same. You're not multiplying it, you're not changing it, you're just getting an application method. And that's what an extract is. Compost extract is just washing the microbes off the substrate so that you can spray them out.
Mhm. And Chris, are you finding the IPMO is working with things like aphids and other bugs as well, or is it more just like beetle kind of bugs and hard-shelled bugs?
So I have been told by people dealing with it that they're dealing with spider mites, they're
Mm.
dealing with aphids. I believe that any that control with aphids comes from the plant health.
Mhm.
Basically, what the cell wall of the plant is doing is instead of being thin with a bunch of sweet juice in the middle of the cell, a bunch of sweet, cytoplasm, in the center, and a thin cell wall, a plant with microbes present on the level we're talking about increasing with IMO is now getting on-demand minor nutrients that enables it to express its full genetic potential, and the cell wall becomes this thick layer of fats and proteins.
Mm.
It becomes a robust cell wall rather than a thin cell wall with a bunch of sweet juice in the middle, like a Capri Sun. Stab the little bugs stab their little feeder bit in, and they suck out all that sweet juice, and they didn't have to work very hard to get through that cell wall. Now, a healthy plant has this thick cell wall, and those aphids can't eat the fats and proteins. So they have to burrow through, and they spend all this energy to get to that inside sweet stuff. And that work actually decreases their population because their food source is basically taking them a week to harvest instead of a day to harvest, if that makes sense. I mean,
Yeah,
respectively for us. So,
yeah.
um, I don't know if IPMO is on application effective against aphids, but IMO is extremely effective against aphids. So spraying liquid IMO is one of the protocols in an aphid susceptible crop.
Okay, awesome. And this it sounds like this might be a bit of a clarifying question. Um, Greg says Japanese beetles are taking over the countryside in recent years. How do we go about creating an infected, dead Japanese beetle? Would it work to add Bavarian? I don't know how to say
Bavaria bassiana.
Bastian.
So
Thank you.
yeah,
And because he says he hasn't seen infected beetles, only just dead. So I guess I'm wondering, can you use just regular healthy beetles, or do you need to find infected beetles, and can you use dead ones?
great question. My video goes into this. I literally take you through the process of using the beetles, how I harvest them, how I get them so they're not crawling out of my rice while I'm trying to cook it. Um, I throw them in the freezer, just a little Ziploc in the freezer for 24 hours, and then they're frozen and not crawling out. So yes, dead because the live beetles don't want to stay there. It's kind of they fly too, so it can be.
Hmm.
I just watched three of your faces go. "Ugh." That's wonderful. Uh, um, so yes, Bavaria bassiana is a naturally is a patented entomopathogenic fungi. Great reference. Yes, Bavaria bassiana is great. Also really expensive, and if you multiply it in on your farm after you bought it from them, they can sue you for using it. However, Bavaria bassiana is naturally occurring everywhere. It's one of those microbes that we found strains of it all over the place. And similar species are also all over the place. What we're talking about is instead of saying that one person that found bloom on that beetle that was effective in eating it, now we're going to buy their product for it's not cheap. We're going to take that same principle, we're going to take our beetle out. We're going to wild craft our own Bavaria bassiana, though we're not going to call it that because they somehow have patented nature. We're going to go out and say we just are using plant-based compost and getting effective kill of our beetles. So no, you do not need to find an infected beetle, though if you do find an infected beetle, you can grow that out and use that on your farm. What we want to do is take what we're talking about doing is taking uninfected beetles, putting them on rice, going out to these wild places and getting them their bodies infected, and then using that to spray our farm.
Awesome.
And it
Thank you.
works. Yeah.
So cool. Are there best times of the year to collect IMOs, or is it a good idea to culture seasonally and combine them? And second part, I'll answer that first, and then we'll go to the next one.
Yeah, if you can, the absolute ideal is to get three collections at least 3 collections a season, 4 seasons a year. A lot of places you can't collect in the winter, so 3 seasons a year. So that would be 9 collections. Now I'm making with 9 different collections my IMO 3 and 4, or I'm taking 9 little bits, spoonfuls of 9 different IMO 2s and making my liquid IMO. That would be ideal because different microbes rise and fall in population at different times a year, and we can culture different ones.
And also,
And
sorry, go ahead.
the other part of that question was important, but I lost it.
Um, the best times of year to collect them, and should we collect them seasonally? Yeah, answered that. The other part is, if our region is mostly coniferous forest, is it good and okay to collect from below the cedar and fir trees, even though they're known to be antimicrobial?
Yeah, so cedar and fir trees are resistant to microbes. They aren't antimicrobial. Cedar and fir trees are so high in carbon, low in nitrogen, that really fungi can't do much with them or their foliage. They're drop down pieces of wood. It's only some things can use them. Redwood is completely antimicrobial. There's nothing wrong with collecting in fir and whatnot, but plains, shrubland, all the different places that you can find fungi visibly are all important. You don't want only forest if you can find other places.
Which makes a lot of sense because if you're in yeah, if you're in like marshlands and you don't have the forest, you're going to the marsh to find your fungi. If you're in the prairies, you're going to well, yeah, the duck, not the sloughs, but we call them sloughs. They're not sloughs, but they're little groves of
Yeah,
trees. That's where you're going to collect your fungi. Yeah, yeah, amazing.
ab- absolutely.
Yeah, and anything else that's really on your heart about IPMOs that we haven't covered, Chris? I know we've gone to a lot of places in this conversation, and I think it's been so awesome, both foundationally and a little and in depth for people who are more familiar. So thank you so much for your insights.
Yeah, it was really easy to have this conversation, and I look forward to more. And even people asking questions directly is welcome. I think just the encouragement to really follow a recipe and try something, even if it's JMS, and really, I'm a big proponent of use what works. So give it a try, and if it's effective, then it's worth the work to do it. And if it's not effective, of course, it's not worth it. So I don't do natural farming because I have some bias to this being the way I do natural farming, because in my long journey of searching on how better to farm profitably for our family farm, this was the elegant method that fit, and this works all over the world. So yeah, I guess just don't drink the Kool-Aid, remain skeptical, and go give it a try, and do a control. Don't apply it on everything. Leave some off, and then if it's you have a 20%, 40% increase, then it's like, oh wow, I could spend some time dialing this in rather
Mhm.
than I'm going to do it because Chris said it was a good idea. Don't take my word
Right,
for it.
right. And Chris, are you familiar with the cow pat pit from biodynamic farming?
I
CP.
am. Yeah, the
How's
horn, the buried horn.
well, the cow pat pit's a bit different.
Oh,
So
sorry.
yeah, it's no, that's okay. Someone just asked a question about it, so I was wondering if you're familiar. But I can answer that a little bit more than it's different. I guess it's similar, but it's quite different than the cow pat pit, Katrina, in terms of like the cow pat pit is you're stirring manure and then you're putting um different I guess it is kind of similar actually, but different. You're putting in different preparations into that cow pit and cow pat pit with the manure that's been aerated, and then you leave that sit for 6 months, and then it comes out with this beautiful microbial rich
Hmm.
fungal yumminess. The difference with this though, and is that we're taking the rice into the natural environment, into nature, and we're having the fungi come into that are and the life to come into that rice, and then we're bringing that in and making um solutions and for
Yeah, so what you're doing with the cow pat pit is nuanced or a specific compost on farm with the materials you have on farm, which is wonderful. And so you're growing out what you already have. And that's amazing. And making nitrogen rich, making plant foods is an important part of it. It's also um, a way to get a very plant ready nitrogen. So you're getting food that your plants are going to lap up very vigorously, have a really easy time eating. You're curing manure in the best way possible. So cool biodynamic concept. The difference there is you're not introducing diversity.
well, the cow pat pit though is like you're putting actually five different actual other preparations into that cow pat pit. So there is diversity, but not in the way that you're talking about, Chris, I think.
Yeah. I think one of the I am not highly versed in biodynamics. I'm sorry, but one of the most similar microbial preps for to compare to IMO would be the buried horn.
Mm.
And what you're doing is you're taking the peak of farm diversity, peak of farm activity at the end of your growing season. You're filling the horn with it, and you're burying it below the frost line because this was a Greenland prep, right? This was originated in Greenland. We're copying something they did. And so they buried it below the frost line, which protected that active microbial community from a freeze. And then in the spring, when all your ground has been frozen and the dormancy level of your microbials is at its lowest, you take this thing that never went completely dormant and you seed your farmland. That is going to jumpstart your microbial activity for the spring, which is going to help to cycle nutrients and increase the kind of the growing season of your soil for your plants. And that's really effective for a place that has a freeze because you're protecting the microbes in that manure that was on your soil from freezing and kind of jump starting it. But again, you're not bringing in more diversity. Now, before we started destroying farmland, maybe we didn't need to go find other diversity. Maybe everything that was on your farm was enough. We just needed to hype it up and kind of reinvigorate it. So the manure farm pit is I don't think I said that right, that you're describing. If you already have active life, you're jump starting and reinvigorating to apply out the life you have there. And if
Mm.
you're well established in natural farming, for example, you don't need to keep applying IMO. You're just composting and feeding your soil. So
Mhm.
um, it's not that we need to apply IMO indefinitely. We really just need to have diversity established and then feed it with good compost, and manure can be one of those ways.
So cool and so much to learn. So cool. Thank you. Um, Thank you for your time,
Really
Chris.
amazing. Yeah, really amazing questions and discussion. I appreciate your always preparedness, Natalie, and your insight into the whole of things, bringing together thought and people such a wonderful way.
Deep gratitude to you. Thank you. Thank you. And there's lots of love for you in the chat. People are clapping their hands and saying thank you. And so we'd love to know what your biggest takeaway was from today. If you want to pop that in the chat too, while we are gifting away some door prizes. We'd love to
Cool.
hear what you learned. Chris,